Hello my dear students, I am Dr.Mondakini Soren, again back with a new video. So again today we are going to study class 9 biology, chapter number 5, the fundamental unit of life, part 2. Okay, then let's start. So first of all we are going to see what we are going to cover in this video. So those topics are cell wall and its characteristics, plasma membrane along with its characteristics, transport through plasma membrane, those are passive and active transport, diffusion and osmosis, differences will also be there, endosmosis and exosmosis along with the differences, hypertonic, hypotonic and isotonic solution, endocytosis and exocytosis, and at last we are going to study about plasmolysis. So first of all we are going to see what is cell wall along with its characteristics. So cell wall, as we already studied in our previous video, so cell wall is what it is: a rigid non-living outer covering of plasma membrane, and it also provides shape to a cell. So I think before we already studied the cell wall is already present in plant cell. So animals do not have cell wall, they directly have the plasma membrane as the outer covering. And here in case of plant cell we get cell wall. So it is what it is, a very rigid and non-living covering, okay, and for which the cell it gets a shape. Then there are various list of organisms which have or do not have cell wall, and if they have cell wall, then what type of cell wall they consist of. So likewise it is given, plants have cell wall, as we already know, and what type of cell wall? It is made up of cellulose. Then fungi also consist of cell wall, and that is made up of chitin or chitin also we can tell. Then bacteria also consist of cell wall, and that is made up of peptidoglycan. While in animals and viruses, cell wall is not there, or they do not have cell wall as their outer covering. Okay, now let's move to the next slide. Plasma membrane, or what we tell is cell membrane also. And what are the characteristics? So plasma membrane, as those like animals and viruses, they do not have cell wall. So the outer covering of these two organisms will be what? The plasma membrane, or what we tell as cell membrane. And this cell membrane is very flexible in nature, and it is made up of lipids and proteins. And it is also called as a selectively permeable membrane. So why it is called as selectively permeable membrane? Because it allows the entry and exit of some materials. So entry and exit means it allows some of the materials to move inside the cell through the plasma membrane, and also some of the materials to move outside the cell through the same plasma membrane. Okay, then it prevents movement of some materials. That means this plasma membrane acts as a blockage system. Okay, because some of the materials that are very important or necessary to the cell, it might block some of those materials to go out to the environment, because this contents or living contents are very essential to the cell. So that is also very much helpful to the cell. Then it allows diffusion of substances like carbon dioxide and oxygen. Okay, I think this is very clear to you about the cell wall and the plasma membrane. Okay, then let us move to the next slide. Then what type of transport occurs through the plasma membrane? Okay, so the type of transport are divided into two. One is the passive transport, and other one is the active transport. And Under passive transport occurs diffusion, and other one is osmosis. So I think we are very familiar with these two words about passive and active, and also with diffusion and osmosis. I think we have already studied in the previous classes. Okay, then about diffusion and osmosis and differences between these two, okay, we are going to study later on. Now let's see what is passive transport. So passive transport, we can tell that those transport of molecules or substances that occur from the region of higher concentration to a region of lower concentration. Okay, while in active transport, that is very opposite to passive transport, that is the movement of molecules or substances from a region of lower concentration to a region of higher concentration. Okay, then for this diffusion of osmosis will be the same one, but diffusion is what? In case of either solid, liquid or gases, and in case of osmosis we can tell about water molecules. And osmosis is also again pathway is divided into two types, that is endosmosis and exosmosis. Endo, that means inside, and exo means outside. Okay, that we are going to study later on. Then active transport, it only happens, or this transport only this transport requires energy, and this energy is used in the form of ATP, that is adenosine triphosphate, and I think ATP is generated in the mitochondria of the cell. Okay, now let's move to the next slide. So here I'm going to demonstrate you what is the passive or differences between passive and active transport. Now see, suppose we are taking passive transport. So in case of passive transport, what happens is substances or molecules move from a region of higher concentration to a region of lower concentration. Either it is diffusion or what we tell as osmosis. Okay. Now in case of osmosis, what happens is endosmosis. One is endosmosis, so I'm writing it as E, and exosmosis, so I'm writing it as E and X. Okay, now let's see about passive transport. So suppose I'm taking up part of plasma membrane. Now what happens during passive transport is movement of molecules from a region of higher concentration to lower concentration. Now say molecules from region of high concentration. Suppose the cell, inside the cell there are higher concentration of molecules, okay, inside the cell higher concentration, and outside the cell molecules are of lower concentration. Now what will happen? Movement of molecules from a region of higher concentration to a region of lower concentration through this semi-permeable membrane or selectively permeable membrane. Okay, now this is what passive transport, or we can tell about diffusion and osmosis also. Okay, now what is endosmosis? Endosmosis, endo means inside, so I'm writing it as in, and exo means outside, so I'm writing it as out. Okay, now suppose I'm drawing again a part of plasma membrane, and then here substances or molecules are here, inside and outside. So inside, endo means inside, so from outside molecules or substances will enter inside the cell. Okay, and when molecules or substances will enter inside, then what will happen? Suppose I'm taking a balloon, and then I am pouring some water to it. So what will happen is the balloon will swell. Okay, swell. So that will be in the case of cell also. When molecules enter inside the cell, then what will happen? The cell will swell. And in case of exosmosis, suppose again I am drawing the cell and the outer environment, then what will happen is molecules will move from inside of the cell towards the outer environment, that is out. I am writing the out. So the molecules or substances will move out. And so what will happen when the contents of the cell will move out through this membrane? The cell will ultimately shrink. Shrink means it will get dry up. Okay, so have you seen raising? Yes, it is because the grapes they lose the water content and then totally dry up and then form resin. So this will also be in the case of cell. Okay, I think this is very clear to you. Okay, then let's move to active transport. Now, in case of active transport, so I am writing A and T. What happens is molecules of substances will move from a region of lower concentration to a region of higher concentration. Okay, now let's see. This is the semi-permeable membrane, and suppose this is again the cell contains higher concentration of molecules, and the outside the cell lower concentration. Okay, what will happen is molecules will move from a region of lower concentration to higher concentration. That means in this case what happens is the molecules will move inside the cell, but what is required here is energy in the form of ATP, and ATP is produced where? In the mitochondria of the cell. ATP, that means adenosine triphosphate. Okay, then let's move to the next slide. So the main differences between diffusion and osmosis, let us see. Diffusion means what? Diffusion means movement of substances, either solid, liquid or gas, from a region of higher concentration to a region of lower concentration. Okay, it plays an important role in gaseous exchange of carbon dioxide and oxygen between the cells as well as the cell and its external environment. Then, in case of osmosis, it is the movement of water molecules from a region of lower concentration to a region of higher concentration. And osmosis plays an important role in exchange of solute or solvent between the cells as well as the cell and its external environment. So what is solute and what is solvent? So solute is what? It is a substance that is being dissolved. Okay, for example, we can take a sugar, salt. So it dissolves in water. Okay, so water will be called as what? Solvent, and the salt or sugar that we dissolve in the water, that will be called as solvent. Okay, now let's move to the next one. So endosmosis, as we already studied, exosmosis also we studied that. Endosmosis is what? Movement of solvent into the cell. Okay, so this occurs in case of water molecules. For this reason, we should denote it as either solute or solvent. So endosmosis is movement of solvent into the cell, and exosmosis is what? Movement of solvent outside the cell. Then comes hypertonic, hypotonic and isotonic solution. So what do we mean by these three terms? So hypertonic, hyper, as we already know, hyper means more, isn't it? Hypo, if we tell hypo means less, and iso means so and so, no net change we can tell. Okay, now let us study about these three terms. So first of all, we'll study about what is hypertonic. Hypertonic, now say more solute concentration and less solute solvent. So hypertonic, that means, let me draw our semi-permeable membrane or the plasma membrane. Suppose this is the plasma membrane, and more solute concentration. Suppose the cell contains more solute. Okay, more solute. So I am writing it as MS. And outside the cell, it contains less solvent, LS. So what will happen is the molecules from inside the cell will move outside to the external environment. And already we studied that when contents of the cell move through the plasma membrane to the outer environment, then what happens is the cell will shrink. Okay, now let's move to the next one, hypotonic. Hypo, that means less. So less solute concentration. Hypotonic means less solute concentration and more solvent concentration. So let us again take a part of plasma membrane, and inside the cell there will be less solute concentration, and outside the cell there will be more solvent concentration. Then what will happen? Those molecules from the outer environment will travel inside to the cell, and as a result the cell will swell in size. And in case of isotonic solution, what will happen? If we place a cell, and suppose I am drawing a plasma membrane, and the amount of solute that is present inside the cell and the amount of solvent are suppose same, then what will happen is, so even if the molecules from the outer environment will move inside to the cell, again the same amount will move outside to the external environment. So this will go on repeating, recycling, like so. There will be what? No net change. Like before we got in hypertonic, the cells shrink, the cell will shrink, and in case of hypotonic, the cell will swell. But here no net change will occur. Okay, that means movement, even if the movement will occur, but no net change, because how much will go in, that much will go out also. Okay, now let's move to the next. So again we come to some different terms like endocytosis and exocytosis. So what do we mean by endocytosis and exocytosis? So endo, when we tell endo, that means inside, as I already told, endo means inside, so I am writing here as in. Exo means outside. Okay, out. So in case of amoeba, anyone tell me what is amoeba? Amoeba is a unicellular organism. So in the previous video you can find out, I gave some of the examples like amoeba, chlamydomonas, bacteria, euglena. All those are what? Those are unicellular organism. So unicellular organism. So uni means what? Single cell organism. And amoeba is what? It is a unicellular organism made up of only one cell, and this organism we can find in freshwater ecosystem. And peculiar point for this amoeba, we can tell that it has plasma membrane as the outer covering, and it has false feet, or we can tell pseudopodia, as because its feet. It has false feet because these feet, as you can see in the diagram, these feet are only visible when the amoeba moves or feed on some substances. Okay, now see, what do you mean by endocytosis and exocytosis in case of amoeba? So suppose our food comes near to an amoeba. What happens will this plasma membrane will, or the pseudopodia will stretch its arms, and it will form finger-like finger-like structure, and then this will fuse together, and this food will be carried from the outer environment towards the body of the amoeba. It will enter inside the body of the amoeba, and to the certain point it will move, and then after the amoeba engulf it, what will happen is there will be production of waste materials. Okay, and this toxic or waste materials will be brought out of the cell or out of the body. As it entered the body, those pseudopodia or feet will again this waste material will be brought out of the body, and then these feet will separate, and then it will move the food to the and release in the water body. Okay, so this waste material will be released the water body. So this is what the process of engulfing of food, and then after the digestion process is over, the releasing of the waste material to the water body. So this means what? Endocytosis means and process of engulfing of food, digestion of food by the amoeba, and exocytosis means the release of the waste materials from the body of the amoeba. Okay, so I think this is very clear to you, and it is a very easy topic, Okay, then let's move to the next one. At last, we have the plasmolysis. So what do we mean by plasmolysis? Plasmolysis is a term that is only related to what? Only related to plants. So when a cell is placed in hypertonic solution, what will happen is water moves out of the cell, which leads to the shrinkage of protoplasm. That means what we tell as living contents of the cell away from the cell wall. So in case of plant cells, suppose this is the outer covering or the cell wall, because plant cell has cell wall. So this is the outer covering of the plant cell, and it is called as cell wall. Okay, then inside the cell wall, what we have is the plasma membrane. Okay, plasma membrane. And inside the plasma membrane, we have the nucleus, the Golgi bodies, then ribosome, the vacuoles, then cytoplasm, then mitochondria, etc., etc. Okay, now see, what will happen is these cell organelles along with the cytoplasm that I have drawn. So these all together are called as the living content of the cell. Now, as and when we place the cell in a hypertonic solution, water will move out of the cell to the external environment, and this will lead to shrinkage of the protoplasm. Shrinkage of the protoplasm means these cell contents will move out of the cell, and as a result the cell wall will remain in the outer part, and these living contents will get dried up or it will shrink, and as a result it will move away from the cell wall. You can see how it is moving away from the cell wall. So this process in case of plant cell is called as plasmolysis. I think this is very clear to you. Okay then. Mm, Today we have studied about the cell wall along with its characteristics, then what type of organism consists of cell wall, what type of organisms do not have cell wall, and about the plasma membrane, what it is made up of, and what type of substances gets diffused through the plasma membrane. Okay, then what type of transport occurs through the plasma membrane. Then we got about the endosmosis and then exosmosis, and then difference between diffusion and osmosis. Then about hypertonic, hypotonic, isotonic solution. Okay, then about endocytosis and exocytosis in case of amoeba. Then at last we studied about the plasmolysis in case of plant cell. So I think these points are very clear to you, and then hope will meet very soon, and thank you for watching my video. And if you like my video, please share, comment and subscribe my channel. So stay tuned for the next one.